All right, we'll start off now our second half of the first day of this session. And I have, as always, impressed by the diversity of the presentations, the care that's gone into them, the effort that's made. It certainly reflects so well on the APRES organization. Starting off the mid-morning session will be a paper entitled Development of Diagnostic Tools Against Important Quarantine Viruses of Peanut. And Anushi Deraniyagala is going to be our first presenter.
Hello everyone, I'm Anushi Deraniyagala from University of Georgia Tifton campus. I'm going to present a part of my master's research, Development of Diagnostic Tools Against Quarantine Viruses of Peanut. Peanut is an economically important legume, being a major source of protein and vegetable oil. The United States contributes around 6% of the total world peanut production, while Georgia contributes around 45% of it. Since I work on quarantine pathogens, I would like to introduce the technique or the process called plant quarantine. This ensures disease and pest-free plants. Usually, the plant is isolated and then tests are being carried out to detect the presence of a pest or a pathogen. The importance of this process is this protects the balance of the natural ecosystem present in a country while facilitating the safe international trade of agricultural products. I work on 3 plant quarantine viruses: Groundnut bud necrosis, Orthotospovirus, Indian peanut clump virus, and peanut clump virus. I would like to discuss few facts of these viruses which makes them important quarantine pathogens. I will start with GBNV. This is also known as peanut bud necrosis Orthotospovirus. This belongs to the genus Orthotospovirus, where the type of species is tomato spotted wilt Orthotospovirus, which is present here. And sometimes this can cause a very high yield loss, around 70%, even though management strategies are applied. The major symptoms include stunting, chlorotic spots, and necrosis. The vector is Thrips species. For this genus, GBNV is efficiently transmitted by blossom, chili, and melon thrips. This also has a very wide host range, including leguminous and solanaceous hosts like peanut, potato, bean, and tomato. Why this is important in aspect of quarantine? Because it causes a very high annual loss in Asia where it is currently present. And in some countries like India, when it comes to important crops like tomato, this can cause a very high disease incidence, around 90%, and a disease loss around 80%. The vectors I mentioned earlier all are present within the US. And other than that, this map shows the collective host densities within the United States of peanut, tomato, and potato. So in some states, there is a very high host density, which confirms the fact that GBNV, just in case if introduced to here, there is a high probability to get established. I also work on Indian peanut clump virus and peanut clump virus, which are pet viruses. So Indian peanut clump virus is further identified as 3 different strains, D, H, and L. The major hosts include wheat, sorghum, and maize, and groundnut. It is distributed in the continents of Africa and India. The vector Polymyxa graminis is present within the US, which transfers 2 viruses. And these viruses are very important for winter wheat since they cause a significant yield loss in winter wheat. The other important fact that makes this important in quarantine aspect is this is seed transmitted, which makes this highly difficult to control unless you develop a resistant variety, which takes a huge effort and a good amount of time. With all this information, my objective was to develop a diagnostic tool to detect all these viruses, because as these viruses listed as in important quarantine viruses list recently, there are no efficient diagnostic tools available right now. So next, I would like to go through the methodology I followed, and also I would share some of the results I got. I started with sample collections. Since these are quarantine viruses, I got the samples from outside collaborators. Then, though the symptoms were very clear on the peanut leaf samples infected with these viruses that I got, we still decided to confirm the presence of the virus using molecular diagnostic tools like RT-PCR. So we first designed the primers targeting specific regions in the genome, and then we extracted RNA, because all these viruses are RNA viruses, and then we got the amplifications through RT-PCR, we cloned them, and we sequenced. So finally, we analyzed the sequences with nucleotide blast tool in the GenBank, comparing with the other sequences in the GenBank. These are the viral genomic regions targeted for RT-qPCR. For GBNV, I targeted the regions movement protein and nucleocapsid. For IPCV isolates, I targeted hot protein and triple gene block regions. For PCV, I targeted hot protein and RNA-dependent RNA polymerase regions. Here you can see the applicants that I got for GBNV movement protein. And then this is the result I got when the amplicon of GBNVN protein was blasted. So the sequence of the amplicon successfully aligned with the other similar sequences in the GenBank, confirming the presence of the virus. I followed the same procedure for the other viruses as well. Then the major part of my research, development of nuclear-based diagnostic tool. When it comes to the diagnostic tools available currently, we have chosen to proceed with RT-qPCR because it's highly specific and it also high sensitivity, and also it takes quite less time compared with other tools. RT-qPCR is in two different forms, SYBER-Green and Tachman. So for SYBER-Green, the fluorescent dye SYBER-Green is involved, where it incorporates to double-stranded DNA. So the primers here are the only factors involved in the specificity, while in the Tachman, other than the primers, probe is also involved in the specificity, which makes Tachman is more sensitive, more specific than SYBER-Green. The third step was to develop SYBER-Green-based RT-qPCR. I started with designing primers, RNA extraction, DNA treatment, and finally the standardization. For the primer designing, I targeted the region's movement protein for GBNV, hot protein, and RT for IPCV, and we also included internal control. The amplicon size was around 100 to 250 because it is the amplicon size preferred for RT-qPCR, which is quite low than RT-PCR. Then first, we optimized annealing temperature for primers. For that, we used the temperature gradient. So then based on the CQ value, we decided what is the best temperature to carry out the RT-qPCR. So this is the results I have gained for GBNV movement protein, and based on the CQ value, we decided to proceed with 60 Celsius as the annealing temperature, and this is the finalized protocol for RT-qPCR. Other than after this standardization of annealing temperature, we move to check the specificity, which is very important because this primer should be specific to this virus. For that, we included RNA from GBNV infected plant, and then we included RNA from TSW infected plant, and then we included the control RNA from healthy plant. The amplification of the viral gene was observed in the RNA from GBNV infected plant, which is the sample 1. And we didn't observe any amplification for the other RNA samples, which confirms the idea that this primers are pretty specific. Then we proceeded to check the sensitivity, which is also another important aspect. Here we used diluted concentrations of total RNA, and the results shown here are movement protein of GBNV. We were able to detect the virus in a total RNA of 0.1 nanogram per microliter, which is very low. Then this is the results. This is the optimized RT-qPCR protocol for amplification of RDRP gene, and the results shows the RDRP gene of IPCV de-amplification. The final step is to develop Tachman RT-qPCR, which involves the designing of primers and probes first, and then RNA extraction, and finally the standardization. So these are the 3 regions targeted. Well, the 2 regions targeted for the virus and the control, internal control. And we used 3 different dyes for the probe. So this is the results I got. So these 3 graphs are the amplification of these particular regions through temperature gradient. This is for movement protein, and this is for nucleocapsid protein, and this is for the internal control. So after seeing these graphs, we developed multiplex RT-qPCR. So here we add all these primers together in one tube, which makes the process quick and which also helpful in saving reagents as well. So successfully we could able to detect these three curves without any cross interactions. So with that, here comes the end of my presentation. As a conclusion, SYBER-Green-based RT-qPCR and Tachman-based RT-qPCR are developed for GBNV, and these techniques of these SYBER-Green-based and Tachman-based RT-qPCRs are currently being developed for the viruses PCV and IPCV. And finally, we hope to transfer this technology for validation and verification by APHIS and ICRISAT. With that, I would like to thank my committee members, Dr. Sudeep Bag, my major advisor, Dr. Kulbert, and Dr. Dium. I would also like to thank my lab members, collaborators, and the funding agencies. Thank you all.
Thank you. And we have time for questions. And our first question comes from Dr. Barry Tillman. And what is the timeline for transfer to APHIS? What is the timeline for transfer to APHIS?
APHIS timeline, I hope the timeline means the APHIS the stage of the life cycle of the APHIS.
No, I think the question is, how long do you envision it going before you're able to transfer this technology, transfer this data, transfer this methodology to APHIS so they can use it?
Okay, so well, for GBNV, we have optimized almost all the things. So we hope it would take like 2 months to transfer GBNV, but to transfer the methodologies related to PCV and IPCV would take some time, maybe 6 months from now.
Okay. An additional question is, how do you see this being deployed? Is just when we suspect that the viruses are here, or we start screening prior to arrival, or how do you see your technology being deployed to protect the peanut farmers in America?
Well, there are, like, sometimes the breeding materials are being imported. So the breeding materials, we are planning to implement this technology to check the breeding materials so that we don't take any breeding materials with the virus in them, because formerly it has been reported that some viruses started to settle down here, or else they were introduced to here because of the breeding materials.
All right, excellent. And that's the time we have for questions. And so thank you very much.